This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Hello and welcome to our deep dive into one of the most fascinating areas of origin of life research, the RNA world hypothesis. I'm excited to take you on a journey exploring how life may have first emerged on our planet over 4 billion years ago. We'll be looking at cutting-edge scientific research that is shedding light on the potential role of RNA in the earliest stages of life on Earth. For those who may be unfamiliar, RNA stands for ribonucleic acid. It's a molecule similar to DNA that plays crucial roles in biological processes. The RNA world hypothesis proposes that early forms of life relied on RNA to serve as both the genetic material and the catalysts for chemical reactions, before the emergence of DNA and proteins that dominate life as we know it today. Over the next hour or so, we'll explore several key areas of research related to the RNA world. We'll look at how RNA molecules may have first formed on the early Earth, how they could have developed the ability to self-replicate and catalyze reactions, and how they may have eventually given rise to the DNA and protein-based life we see today. We'll also touch on how RNA research is providing insights into the search for life beyond Earth and enabling exciting new biotechnology applications. My goal is to make this complex scientific topic accessible and engaging for both science enthusiasts and those who may be new to the subject. We won't be diving into the technical details of specific studies, but rather focusing on the big picture ideas and implications. By the end, I hope you'll have a solid grasp of the RNA world hypothesis and why it's such an active and important area of research. So whether you're a student, scientist, or simply someone curious about the origins of life, I invite you to sit back and join me on this fascinating scientific journey. Let's dive in. Topic 1. Origins of RNA and early Earth chemistry. To understand how RNA may have played a central role in the emergence of life, we first need to consider how RNA molecules themselves could have formed on the early Earth. This takes us back over 4 billion years to a time when our planet was very different from today, a world with no oxygen in the atmosphere, no ozone layer, and likely frequent bombardments from comets and asteroids. Research in prebiotic chemistry is working to recreate the conditions of early Earth and understand how the building blocks of RNA could have spontaneously formed from simpler molecules. One of the key challenges is that RNA is a relatively complex molecule made up of 4 different nucleotide bases: adenine, guanine, cytosine, and uracil, as well as ribose sugar and phosphate groups. Recent experiments have shown promising routes for how these components may have been synthesized through chemical reactions in primordial pools, aided by energy sources like UV light from the young sun. For example, formaldehyde, a simple organic compound likely present on early Earth, can react to form ribose sugars. Nucleobases have been successfully synthesized from hydrogen cyanide and other small molecules under conditions mimicking those thought to exist billions of years ago. However, getting from individual building blocks to full RNA molecules presents another set of challenges. This is where minerals and clays may have played a crucial role. Experiments have shown that certain clay minerals, like montmorillonite, can catalyze the polymerization of nucleotides into short RNA strands. The charged surfaces of these minerals may have provided a scaffold to concentrate and organize RNA monomers, facilitating their linking into longer chains. Interestingly, some researchers have proposed that RNA may not have been the very first genetic polymer. Other nucleic acid-like molecules with simpler backbones, sometimes called pre-RNA world molecules, may have preceded RNA. Potential candidates include peptide nucleic acids, PNA, threose nucleic acid, TNA, or glycol nucleic acid, GNA. These alternative genetic polymers are being studied for their ability to store information and potentially replicate in prebiotic conditions. The study of meteorites and interplanetary dust particles is also providing clues about the organic compounds that may have been delivered to early Earth from space. Some meteorites contain nucleobases and other organic molecules, suggesting that some of the raw materials for RNA may have had an extraterrestrial origin. All of these lines of research are gradually filling in the picture of how the molecular building blocks for an RNA world could have arisen through abiotic processes on the early Earth. While many details remain to be worked out, the general outline of a pathway from simple organic molecules to complex RNA polymers is beginning to take shape. This origin story sets the stage for the next crucial step: how these early RNA molecules could have developed the ability to catalyze chemical reactions and self-replicate, laying the groundwork for the emergence of life. And that's what we'll explore next. Topic 2. RNA catalysis and the first ribozymes. One of the key features that makes RNA a strong candidate for the origin of life is its ability to act as both an information carrier and a catalyst for chemical reactions. This dual nature of RNA was not always appreciated. For many years, it was thought that only proteins could act as biological catalysts or enzymes. However, a groundbreaking discovery in the 1980s changed this view dramatically. Researchers found that certain RNA molecules could catalyze specific chemical reactions, including the cleavage and joining of other RNA molecules. These catalytic RNAs were dubbed ribozymes, a portmanteau of ribonucleic acid and enzyme. This discovery was revolutionary because it showed that RNA could potentially carry out many of the functions necessary for early life, without the need for proteins. In modern cells, protein enzymes carry out most catalytic functions, while DNA stores genetic information and RNA acts primarily as an intermediary. But in a hypothetical RNA world, RNA could have played all of these roles. Since the initial discovery, many different types of ribozymes have been identified in nature and created in the lab. Some of the most well-known natural ribozymes include: 1. The ribosome. The cellular machine that synthesizes proteins is actually a ribozyme at its core. While the ribosome contains many proteins, the actual peptide bond formation is catalyzed by the RNA component. 2. Self-splicing introns. These are segments of RNA that can cut themselves out of larger RNA molecules and join the remaining pieces back together. 3. The RNA component of RNase P. This ribozyme is involved in processing transfer RNAs in modern cells. In addition to these naturally occurring ribozymes, researchers have used techniques like in vitro evolution to create artificial ribozymes with a wide range of catalytic abilities. These include RNAs that can catalyze the formation of peptide bonds, linking amino acids together, perform simple metabolic reactions, and even catalyze the copying of other RNA molecules. The ability of RNA to act as a catalyst is crucial for the RNA world hypothesis because it provides a potential mechanism for self-replication. A self-replicating RNA would need to be able to make copies of itself without the aid of proteins. While no naturally occurring self-replicating RNA has been found, researchers have made significant progress in creating artificial systems that approach this capability. For example, some engineered ribozymes can catalyze the formation of short segments of complementary RNA. While these are not yet capable of fully copying themselves, they demonstrate the potential for RNA-based replication systems. Other experiments have shown how networks of ribozymes could potentially work together to copy each other, providing a model for how more complex replication systems might have evolved. The catalytic versatility of RNA also suggests how early metabolic processes could have been carried out in an RNA world. Simple ribozymes might have catalyzed the synthesis of important molecules, the harvesting of energy from chemical gradients, or the construction of primitive membranes. As we imagine the transition from non-living chemistry to the first forms of life, ribozymes provide a plausible mechanism for how this leap might have occurred. A population of self-replicating, catalytic RNAs could potentially undergo natural selection and evolution, gradually increasing in complexity and capability. This brings us to an important question. If RNA was indeed the original basis for life, how did we get from an RNA world to the DNA and protein world we see in all life today? That's what we'll explore in our next topic. Topic 3. Transition from RNA world to DNA, protein world. The RNA world hypothesis provides a compelling scenario for the origins of life, but it also raises an important question. If life began with RNA, how did we end up with the DNA and protein-based life we see today? This transition represents a major evolutionary milestone, and understanding how it might have occurred is a key area of research. Let's start with the emergence of DNA. DNA, deoxyribonucleic acid, is chemically very similar to RNA, with a few key differences. The most significant is that DNA uses deoxyribose sugar in its backbone instead of ribose, making it more stable and less prone to hydrolysis. This stability makes DNA a better long-term storage medium for genetic information. Researchers have proposed several scenarios for how DNA might have evolved from RNA. 1. Some hypothesize that DNA initially evolved as a way to store genetic information more stably, perhaps in response to environmental pressures like UV radiation or high temperatures. 2. Others suggest that DNA might have originally evolved as a kind of parasite, a molecule that was accidentally replicated by RNA machinery and then took on a functional role. 3. Another possibility is that DNA emerged as a byproduct of RNA metabolism and was later co-opted for information storage. Regardless of the exact mechanism, the transition to DNA would have required the evolution of enzymes to synthesize DNA from RNA templates and to replicate DNA. In modern cells, these functions are carried out by proteins, but they may have originally been performed by ribozymes. The evolution of protein synthesis represents another major transition. In all modern life, proteins are synthesized by ribosomes, which are themselves complex RNA protein machines. Interestingly, the core function of the ribosome, the formation of peptide bonds between amino acids, is carried out by the RNA component, suggesting that protein synthesis may have its roots in the RNA world. The development of the genetic code, the system by which nucleotide sequences are translated into amino acid sequences, was a crucial step in this transition. Researchers have proposed various models for how the code might have evolved, often involving RNA molecules that could bind to both amino acids and specific nucleotide sequences. As protein synthesis became more sophisticated, proteins would have been able to take over many catalytic and structural roles from RNA. Proteins are generally more efficient catalysts than ribozymes and can perform a wider range of chemical reactions. This would have allowed for the development of more complex metabolic processes and cellular structures. However, it's important to note that this transition was likely gradual, with RNA and proteins coexisting and co-evolving for a long time. Even in modern cells, RNA continues to play crucial roles beyond just carrying genetic information. The shift from RNA to DNA as the primary genetic material would have brought several advantages. 1. Increased stability of genetic information. 2. Separation of information storage, DNA, from information processing, RNA and proteins. 3. Potential for larger genomes and more complex organisms. This transition also laid the groundwork for the development of more complex cellular processes, including the evolution of the nucleus in eukaryotic cells and sophisticated regulatory mechanisms. While many details of this transition remain to be worked out, the general outline of a path from an RNA world to a DNA and protein world is becoming clearer. This model provides a framework for understanding one of the most fundamental transitions in the history of life on Earth. In our next topic, we'll explore how remnants of the RNA world persist in modern organisms and how studying RNA in today's life forms can provide insights into its ancient roles. Topic 4. RNA structure and function in modern organisms. While the RNA world hypothesis focuses on the role of RNA in the origins of life, studying the diverse functions of RNA in modern organisms provides valuable insights into its ancient roles and evolutionary history. Despite the dominance of DNA and proteins in contemporary life, RNA continues to play crucial and varied roles in all living systems. Let's start by considering the diversity of RNA types found in modern cells. 1. Messenger RNA, mRNA. These molecules carry genetic information from DNA to the ribosome for protein synthesis. 2. Transfer RNA, tRNA. These small, highly structured RNAs bring specific amino acids to the ribosome during protein synthesis. 3. Ribosomal RNA, rRNA. As we discussed earlier, rRNA forms the core of the ribosome and catalyzes peptide bond formation. 4. Small nuclear RNAs, snRNAs. These are involved in splicing pre-mRNA molecules. 5. Small nucleolar RNAs, snRNAs. These guide chemical modifications of other RNAs. 6. MicroRNAs, mRNAs, and small interfering RNAs, siRNAs. These regulate gene expression by binding to mRNAs and either blocking their translation or causing their degradation. 7. Long non-coding RNAs, lncRNAs. These have various regulatory functions, including control of gene expression and chromosome structure. This diversity of RNA types and functions in modern cells suggests that RNA has continued to evolve and take on new roles long after the hypothetical RNA world. Many of these functions involve complex interactions between RNA and proteins, highlighting the co-evolution of these two types of biomolecules. One particularly interesting area of research is the study of riboswitches. These are segments of mRNA that can change their structure in response to small molecules, thereby regulating gene expression. Riboswitches are found in many bacteria and some eukaryotes, and they provide a mechanism for cells to respond to their environment without the need for proteins. Some researchers have suggested that riboswitches might be molecular fossils from the RNA world. Another important area of study is RNA modifications. Just like DNA and proteins, RNA molecules can be chemically modified after they're synthesized. These modifications can affect the RNA's structure, stability, and function. Over 100 different types of RNA modifications have been identified, forming what's sometimes called the epitranscriptome. Some of these modifications are highly conserved across all domains of life, suggesting ancient origins. The structure of RNA molecules is also a rich area of study. Unlike DNA, which typically forms a stable double helix, RNA can fold into complex three-dimensional structures. These structures are often crucial for the RNA's function. For example, the complex folding of rRNA in the ribosome creates the active site for peptide bond formation. Studying RNA structure and function in modern organisms has led to some surprising discoveries. For instance, many regions of the genome once thought to be junk DNA are now known to be transcribed into functional non-coding RNAs. This has led to a re-evaluation of our understanding of genome function and evolution. RNA also plays crucial roles in many viruses. Some viruses, like influenza and coronavirus, use RNA as their genetic material instead of DNA. Studying these RNA viruses provides insights into alternative strategies for information storage and replication, and may offer clues about the capabilities of early RNA-based life forms. The central role of RNA in protein synthesis in all living things is perhaps the strongest piece of evidence for the RNA world hypothesis. The fact that the ribosome, the universal cellular machine for making proteins, is at its core a ribozyme, suggests that protein synthesis evolved in a world where RNA was the predominant biopolymer. All of these modern functions of RNA, from carrying genetic information to regulating gene expression to catalyzing chemical reactions, showcase the versatility of this molecule. This versatility is what makes RNA such a compelling candidate for the original molecule of life. In our next topic, we'll explore how RNA might have made the leap from individual molecules to the first cellular-like structures, a crucial step in the origin of life. Topic 5. RNA and the origins of cellular life. As we've explored the potential roles of RNA in early life, we've mainly focused on individual molecules. But life as we know it exists in cells, compartmentalized structures that separate the internal environment from the external world. So how might RNA-based life have made the transition from free-floating molecules to something resembling primitive cells? This question brings us to the concept of protocells. Protocells are simple, self-organizing structures that are thought to represent a step between non-living chemistry and the first true cells. They typically consist of a membrane-bound compartment containing some kind of information-carrying molecule, like RNA. The formation of these early compartments is a crucial area of study in origin of life research. One leading hypothesis is that fatty acids, which can spontaneously form bilayer membranes in water, may have created the first cellular compartments. These simple membranes could have encapsulated RNA molecules, creating isolated environments where early biochemical processes could occur. Experiments have shown that simple protocells can be created in the laboratory using fatty acid membranes and RNA. These protocells can grow, divide, and even compete with each other for resources, demonstrating some of the basic properties we associate with life. But how might RNA have functioned within these early compartments? Several possibilities have been proposed. 1. Catalytic RNAs, ribozymes, within the protocell could have carried out simple metabolic reactions, generating molecules necessary for growth and replication. 2. RNA could have played a role in regulating the composition of the protocell. For example, certain RNA sequences might have interacted with the membrane, influencing its permeability or stability. 3. Self-replicating RNA systems within the protocell could have provided a mechanism for heredity and evolution. One of the challenges in this field is understanding how these early compartments could have acquired the materials needed for growth and replication. Recent research has shown that simple protocells can selectively take up certain molecules from their environment, providing a potential mechanism for nutrient acquisition. Another important question is how these early compartments might have divided. In modern cells, division is a complex process involving many proteins. However, researchers have demonstrated that simple protocells can divide spontaneously under certain conditions, driven by physical forces. The energetics of early cellular life is another crucial area of study. In modern cells, ATP, adenosine triphosphate, serves as the primary energy currency. Interestingly, ATP contains an RNA nucleotide, leading some researchers to speculate that it might be a molecular fossil from the RNA world. Early protocells might have used simpler energy systems, perhaps based on chemical gradients across their membranes. As these protocells became more complex, they would have faced new challenges. For example, as RNA genomes grew larger, mechanisms would have been needed to ensure accurate replication and distribution to daughter cells. This might have driven the evolution of more sophisticated cellular structures and division mechanisms. The transition from simple protocells to the complex cells we see today was undoubtedly a long and gradual process. It likely involved the co-evolution of genetic materials, RNA and later DNA, functional molecules, like ribozymes and later proteins, and cellular structures, membranes and other organelles. Understanding this transition is not just of historical interest. Research into protocells and minimal cells has potential applications in synthetic biology and nanotechnology. For example, engineered protocells might be used for drug delivery or as simplified models for studying cellular processes. Moreover, this research has implications for the search for life beyond Earth. If we understand the minimal requirements for cellular life, it can guide our search for biosignatures on other planets and moons. The study of RNA in the context of early cellular life brings together multiple disciplines, including chemistry, physics, and biology. It requires us to think about life not just in terms of individual molecules, but as complex, self-organizing systems. In our next topic, we'll explore some of the experimental approaches scientists are using to test hypotheses about the RNA world and early cellular life. Topic 6. Experimental approaches to studying the RNA world. Studying events that occurred billions of years ago presents unique challenges. We can't directly observe the origin of life or the early RNA world. Instead, researchers use a variety of clever experimental approaches to test hypotheses and gain insights into these ancient processes. Let's explore some of the key methods being used in this field. One of the most powerful techniques is known as in vitro evolution. This approach allows researchers to create large pools of random RNA sequences and then select for molecules with specific properties. Here's how it typically works. 1. A large library of random RNA sequences is generated. 2. The RNAs are subjected to a selection pressure, for example, the ability to bind a specific molecule or catalyze a particular reaction. 3. The most successful RNA molecules are amplified and mutated. 4. The process is repeated multiple times, mimicking natural selection. This technique has led to the discovery of RNA molecules with a wide range of catalytic abilities, some of which might resemble ancient ribozymes. It's also been used to study how complex functions might have evolved from simpler precursors. Another important experimental approach involves recreating the conditions of early Earth in the laboratory. This includes synthesizing prebiotic molecules under conditions that might have existed billions of years ago. For example, researchers have used mixtures of simple chemicals, along with sources of energy like UV light or electrical discharges, to produce organic molecules including the building blocks of RNA. These prebiotic chemistry experiments help us understand how the raw materials for an RNA world might have been generated. They've shown, for instance, how nucleotides, the building blocks of RNA, could have formed from simpler precursors under plausible early Earth conditions. Researchers are also using microfluidic devices to study the behavior of protocells and simple RNA-based systems. These devices allow for precise control over tiny volumes of liquids, mimicking the microscale environments where early cellular life might have emerged. They can be used to study things like protocell division, RNA replication, and simple forms of molecular evolution. Synthetic biology is another powerful approach in this field. By engineering simplified cellular systems or artificial protocells, researchers can test hypotheses about the minimal requirements for life. This includes creating cells with synthetic genomes or engineering protocells that can grow, divide, and evolve. Computer simulations and theoretical models also play a crucial role. These can be used to explore scenarios that are difficult or impossible to test experimentally. For example, simulations can model the behavior of RNA molecules over long time scales or under extreme conditions. They can also help in understanding complex systems, like networks of interacting ribozymes. Another interesting approach involves studying molecular fossils, features of modern biochemistry that might be remnants of the RNA world. The ribosome, with its RNA-based catalytic core, is often cited as an example. By studying the structure and function of these molecular fossils, researchers hope to gain insights into ancient RNA-based systems. Comparative genomics is also providing valuable insights. By comparing the genomes of diverse organisms, including ancient lineages like archaea, researchers can infer information about early forms of life. This includes identifying highly conserved RNA molecules and structures that might date back to the RNA world. It's worth noting that many of these approaches are complementary and are often used in combination. For example, a computational model might predict a certain RNA structure, which could then be tested experimentally using in vitro evolution. These experimental approaches are continually evolving, with new technologies opening up new possibilities. For instance, recent advances in high-throughput sequencing have revolutionized our ability to analyze large pools of RNA molecules. Similarly, improvements in analytical techniques are allowing for ever more precise recreations of prebiotic chemistry. While each of these approaches has its limitations, together they are helping to build an increasingly detailed picture of how life might have emerged from non-living chemistry. They're also generating insights and technologies that have applications far beyond origin of life research, in fields ranging from medicine to materials science. In our next topic, we'll explore how RNA world research is contributing to the search for life beyond Earth. Topic 7. RNA in astrobiology and the search for life. The study of the RNA world and the origin of life on Earth has important implications for the search for life elsewhere in the universe. As we develop a better understanding of how life might have arisen on our planet, we can apply this knowledge to the search for potential biosignatures on other worlds. Let's explore how RNA research is influencing the field of astrobiology. One of the key questions in astrobiology is, what should we look for as signs of life on other planets or moons? Traditionally, the search for extraterrestrial life has focused on water, organic molecules, and certain atmospheric gases like oxygen. However, our understanding of the RNA world suggests that we might need to broaden our search criteria. RNA molecules themselves could potentially serve as biosignatures. While individual nucleotides might form abiotically, Long, complex RNA molecules are unlikely to arise without biological processes. Therefore, detecting such molecules could be a strong indicator of life. However, identifying specific RNA sequences on another planet would be extremely challenging with current technology. Instead, astrobiologists are considering indirect signs of RNA-based life. For example, 1. Certain patterns of chemical disequilibrium might indicate the presence of self-replicating RNA systems. 2. The presence of ribose or other RNA components in specific ratios or concentrations could suggest biological activity. 3. Isotopic fractionation patterns characteristic of RNA-based metabolic processes could be detectable. The study of extreme environments on Earth, informed by RNA World Research, is also influencing our search for life elsewhere. We now know that life can exist in a wide range of harsh conditions, from deep-sea hydrothermal vents to Antarctic dry valleys. This has expanded our concept of the habitable zone, the region around a star where conditions might support life. Moreover, research into the stability and function of RNA under different conditions is helping to define the physical and chemical limits for life. This includes studying how RNA behaves at high temperatures, in highly acidic or alkaline environments, or in the presence of various salts or organic solvents. Such research helps us understand where in the solar system, or beyond, RNA-based life might be possible. For example, some researchers have studied the behavior of RNA in ammonia-water mixtures, which might exist beneath the surface of icy moons like Saturn's Titan. While Earth life is water-based, other solvents could potentially support different forms of life, and RNA might be capable of functioning in such exotic environments. The possibility of shadow biospheres, alternative forms of life coexisting with familiar life on Earth, is another intriguing idea influenced by RNA World Research. If life emerged multiple times on Earth, perhaps based on different chemical systems, it might have done so elsewhere as well. This suggests we should keep an open mind about what constitutes a sign of life. RNA World Research is also informing discussions about the potential for panspermia, the hypothesis that life could spread between planets via meteorites or other means. Understanding the conditions under which RNA-based life might have originated helps us assess the plausibility of life surviving interplanetary travel. As we send probes to other worlds in our solar system, RNA research is influencing the design of life detection experiments. For instance, future missions to Mars or the icy moons of Jupiter and Saturn might include instruments capable of detecting RNA or its components. Beyond our solar system, the study of exoplanets is another area where RNA World Research is relevant. As we discover more Earth-like planets orbiting other stars, understanding the conditions necessary for the emergence of RNA-based life helps us assess their potential habitability. It's important to note that while RNA-based life is one possibility, alien life might be based on entirely different chemical systems. The study of alternative genetic polymers in origin of life research is encouraging astrobiologists to consider a wide range of possibilities. Lastly, RNA World Research contributes to one of the biggest questions in astrobiology. How likely is the emergence of life? If the path from chemistry to biology that we're uncovering on Earth is relatively straightforward, it might suggest that life could be common in the universe. On the other hand, if it required a highly improbable series of events, life might be exceedingly rare. While we're still far from answering these grand questions, the ongoing research into the RNA world and the origin of life is providing valuable tools and perspectives for the search for life beyond Earth. It's an exciting time in this field, with new discoveries on Earth continually informing our exploration of the cosmos. In our final topic, we'll look at how insights from RNA World Research are being applied in modern biotechnology and medicine. Topic 8. Modern applications of RNA research. While much of our discussion has focused on the ancient past, research into RNA and its capabilities is also driving exciting developments in present-day science and technology. The insights gained from studying the RNA world are finding applications in fields ranging from medicine to materials science. Let's explore some of these cutting-edge applications. One of the most prominent areas where RNA research is making a big impact is in medicine, particularly in the development of RNA-based therapeutics. This field has seen explosive growth in recent years, with several notable successes. 1. mRNA vaccines. The COVID-19 pandemic brought mRNA vaccines into the spotlight, but this technology has been in development for many years. These vaccines use mRNA to instruct cells to produce specific proteins, triggering an immune response. This approach, which builds on our understanding of RNA's role in protein synthesis, offers several advantages over traditional vaccines, including faster development times and easier manufacturing. 2. RNA interference, RNA therapies. This approach uses small RNA molecules to selectively silence specific genes. It's based on a natural cellular process discovered through basic RNA research. RNA therapies are being developed for a wide range of conditions, from genetic disorders to cancers. 3. Antisense oligonucleotides. These are short, synthetic RNA or DNA molecules designed to bind to specific mRNA molecules and alter their processing or translation. Several antisense drugs have been approved for treating genetic disorders. 4. Aptamers. These are RNA molecules selected for their ability to bind specific targets, similar to antibodies. They're being developed for both therapeutic and diagnostic applications. Beyond therapeutics, RNA is also being used in diagnostic tools. For example, CRISPR-based diagnostic systems use RNA guides to detect specific DNA or RNA sequences with high sensitivity and specificity. These systems have potential applications in detecting pathogens, identifying cancers, and more. In the field of synthetic biology, insights from the RNA world are informing efforts to create artificial cells and minimal life forms. Researchers are using RNA-based systems to control gene expression in synthetic cells, create logic gates for cellular computation, and even develop self-replicating RNA systems. These efforts not only advance our understanding of the requirements for life but also have potential applications in areas like biomanufacturing and environmental remediation. RNA nanotechnology is another exciting field that draws inspiration from the structural properties of RNA. Researchers are designing and constructing nanoscale structures and devices made from RNA. These have potential applications in drug delivery, molecular computing, and the creation of new materials. In biotechnology, RNA-based tools are becoming increasingly important. CRISPR gene editing systems, which use RNA guides to target specific DNA sequences, have revolutionized genetic engineering. Other RNA-based tools are being used for tasks like controlling gene expression, modifying RNA processing, and even directing the evolution of new biomolecules. The study of ribozymes, catalytic RNAs that played a key role in the RNA world hypothesis, has led to the development of new biocatalysts for industrial and research applications. These include RNA enzymes for RNA synthesis, site-specific RNA cleavage, and even small molecule synthesis. In the field of origins of life research itself, insights from the RNA world are informing efforts to create synthetic life and to understand the fundamental principles of living systems. This work has implications not just for understanding our own origins, but also for the design of novel biological systems and the search for life beyond Earth. Environmental applications are also emerging from RNA research. For example, RNA-based biosensors are being developed to detect pollutants or pathogens in water supplies. RNA-AP tamers are being explored as tools for environmental remediation, capable of binding and potentially neutralizing specific contaminants. In information technology, the information-carrying properties of RNA are inspiring new approaches to data storage and computation. While DNA has received more attention in this area, RNA's greater structural flexibility and catalytic potential offer unique possibilities for molecular computing and data storage. It's worth noting that many of these applications are still in early stages of development, and translating basic research into practical applications often takes many years. However, the rapid progress in fields like mRNA vaccines demonstrates the potential for RNA-based technologies to make significant real-world impacts. Moreover, these applications often feed back into basic research. The challenges encountered in developing RNA therapeutics, for instance, are driving new investigations into RNA stability, cellular uptake, and interaction with biological systems. This creates a virtuous cycle where applied research informs basic science, which in turn enables new applications. As our understanding of RNA continues to grow, it's likely that we'll see even more diverse and impactful applications emerge. The story of RNA, from its potential role in the origin of life billions of years ago to its applications in cutting-edge biotechnology today, is a powerful reminder of how basic scientific research can lead to transformative technologies. As we wrap up our exploration of the RNA world and its implications, let's take a moment to summarize some key points and consider the broader significance of this research. First, the RNA world hypothesis provides a compelling scenario for how life might have emerged from non-living chemistry. The ability of RNA to both carry genetic information and catalyze chemical reactions makes it a strong candidate for the first self-replicating system that could undergo Darwinian evolution. While many details remain to be worked out, the general outline of a pathway from simple organic molecules to complex, RNA-based protocells is becoming clearer. The transition from an RNA world to the DNA and protein world we see today represents a major evolutionary milestone. Understanding this transition not only illuminates our own origins but also provides insights into the fundamental principles of how life operates and evolves. Research into the RNA world is inherently interdisciplinary, bringing together chemistry, biology, physics, geology, and even astronomy. This highlights the complexity of the origin of life question and the need for collaborative, cross-disciplinary approaches in science. The study of RNA in modern organisms continues to reveal new functions and regulatory mechanisms, underscoring the ongoing importance of this molecule in life as we know it. From gene regulation to immune function to development, RNA plays crucial roles that we're only beginning to fully appreciate. This suggests that even after billions of years of evolution, life still bears the imprint of its RNA-based origins. The experimental approaches developed to study the RNA world are not only advancing our understanding of life's origins but also generating new tools and technologies with wide-ranging applications. Techniques like in vitro evolution and the engineering of ribozymes are opening up new possibilities in biotechnology and medicine. RNA world research is also influencing how we think about the possibility of life beyond Earth. By understanding the conditions under which RNA-based life might have emerged and the signatures it might leave behind, we can better focus our search for life on other planets and moons. This research is thus contributing to one of the most profound questions humanity has ever asked. Are we alone in the universe? The rapid development of RNA-based therapeutics, dramatically showcased by mRNA vaccines, demonstrates how insights from basic research can lead to transformative technologies. This underscores the value of fundamental scientific research, even when its practical applications aren't immediately apparent. At the same time, advances in RNA technology raise important ethical and societal questions. As our ability to manipulate RNA grows, we need to carefully consider the implications and potential risks of these powerful new tools. This highlights the need for ongoing dialogue between scientists and the broader public about the directions and applications of this research. The RNA world hypothesis also prompts us to think deeply about what it means to be alive. By exploring the fuzzy boundary between non-living chemistry and living systems, this research challenges us to refine our definitions of life and to consider how the property we call life might have emerged from simpler chemical systems. Looking to the future, RNA research is likely to continue yielding surprises and new possibilities. From new therapeutic strategies to novel materials to artificial life forms, the applications of our growing understanding of RNA seem limited only by our imagination. Moreover, as we uncover more about the potential role of RNA in life's origins, we're gaining insights into the fundamental principles that govern the emergence of complex, self-organizing systems. These principles could have applications far beyond biology, influencing fields like materials science, information technology, and even our understanding of how complex systems like economies or ecosystems function and evolve. In conclusion, the study of the RNA world and its implications is a vibrant, fast-moving field that touches on some of the most fundamental questions in science. How did life begin? How does complexity emerge from simplicity? What are the essential characteristics of living systems? By pursuing these questions, researchers are not only uncovering the secrets of our own origins but also opening up new possibilities for the future. As we've seen throughout this discussion, the story of RNA is one of remarkable versatility and potential. From its possible role in the origin of life billions of years ago to its applications in cutting-edge medical treatments today, RNA continues to surprise and inspire us. It's a powerful reminder of how a single molecule can shape the course of life on Earth and potentially beyond. I hope this exploration has given you a sense of the excitement and importance of this field. Whether you're a student considering a career in science, a researcher in a related field, or simply someone curious about the world around us, there's never been a more exciting time to engage with RNA research and its implications. As we look to the future, it's clear that RNA will continue to play a central role in our understanding of life and in our efforts to address global challenges in health, technology, and environmental sustainability. The RNA world may be ancient history, but its legacy continues to shape our present and our future. Thank you for joining me on this journey through the fascinating world of RNA. I encourage you to continue exploring this topic, to ask questions, and to share your thoughts and ideas. Science is a collaborative endeavor, and who knows, perhaps your curiosity and insights could contribute to the next big breakthrough in our understanding of life's origins and possibilities. If you've enjoyed this video, please consider liking, subscribing, and sharing it with others who might be interested. Your support helps us continue to create content exploring cutting-edge scientific research. And if you have any questions or topics you'd like to see covered in future videos, please leave a comment below. Until next time, keep questioning, keep exploring, and keep marveling at the incredible complexity and beauty of the molecular world that surrounds us and makes us who we are. Thank you for listening, and goodbye for now.